Hello, everyone. This is Rafi. I'm here with my good friend Richard Morris of Two Keto
Hello,
Dudes
Rafi.
and low carb fame more generally, low carb keto fame.
I'm no longer a two keto dude. I'm no longer a keto dude. Well, I
Right,
guess you could call me a keto dude emeritus.
yeah. So you've got Carrie taking over for you.
Right. We're actually the podcast is on hiatus right now because Carl's
Hmm.
working on some other things. But yeah,
Yeah.
Carrie took over for a year
Hmm.
after I left. Yeah.
Okay. Cool. Well, in any case, we were talking about mitochondria
We were.
and what mitochondria are made of, and you are probably the first person then to really uh, describe what mitochondria looks like when simulated,
So
or a patch of it, right?
yeah, I'm just looking at one 15 by 15 nanometer patch of mitochondria of the inner mitochondrial membrane. So it's just one of the
Hmm.
membranes of the mitochondria. So mitochondria are these really cool organelles in our body that give us most of our energy, and we make all of our energy on that inner mitochondrial membrane surface. And so I guess you know that each cell in our body is covered by a plasma membrane. And then inside the plasma membrane, there's the cytosolic environment. And inside the cytosol are organelles like the nucleus and the endoplasmic reticulum and the Golgi apparatus and the mitochondria. And the mitochondria are these little, they're like bean shaped, and they have two membranes. They have an outer membrane that's very smooth, and then they have an inner membrane that's crenellated with all of these sort of invaginations and that's where all the energy is created in our bodies.
Is it kind of
So,
like a brain, which when it looks sort of like squished together, nearly like these
it kind of
folds?
does, or like a walnut, or it's these
Yeah.
little folds, and they call them invaginations, but they're essentially
Hmm.
folds in the inner membrane. The inner membrane is floppy. The outer membrane has like 5% cholesterol. You probably know that the cholesterol molecule, it's a lipid, and it exists in all of our membranes. And in
Hmm.
membranes where there's a lot of the membrane, the cholesterol molecule, like our neuronal membranes, they're quite firm and rigid. In membranes where there's almost no cholesterol molecules, such as the inner mitochondrial membrane has almost none, they're floppy, and they have these pleats. I guess you could say they're like pleats. And at the end of the pleat is where all of the action happens. That's where we
Right.
make all of our energy now, or the majority of our energy in our body. We have other ways to make energy, but the 800-pound gorilla, the one that makes most of our energy, is inside these
Hmm.
mitochondria. And the interesting thing about these is that they have their own DNA. So mitochondria, most of our DNA is in our nucleus. Well, in fact, all of our DNA, all of our genetic material congregates in our nucleus. And when our cells split and divide, the nucleus splits, makes two copies of the DNA, and then it splits, and then you end up with two cells. Well, mitochondria have a different kind of DNA. It's very similar to bacterial DNA. It's sort of a ring of genetic material. And in fact, bacteria have exactly the same mechanism. They have a ring of bacteria instead of these sort of like X-shaped chromosomes that we have. They have just one solid ring. And it is believed that in maybe 3 million years ago in our early evolution, one of our ancestors that wasn't very good at generating energy engulfed a bacteria that was good at generating energy, and they created a symbiosis. And that's how all eukaryotic cells work. We actually have a bunch of little bacteria. It could be 100 of these little things in each of our cells that generate all of our energy. And I've become very fascinated with these things because of their backstory. I mean, the backstory is fascinating. They're pretty close to George Lucas's midi-chlorians. They give us the force, essentially.
Yeah.
So anyway, these so what I've done, I did a biochemistry degree. I did a regular Bachelor of Science in biochemistry in 2.5 years, and then I went into what they call an honours program, which is something in Australia that you can do after your undergraduate. That essentially, I guess in America, if you get a degree and you get summa cum laude, that's essentially the same as getting over 80% in an honours degree in Australia.
Yeah.
So that's what I'm doing, and in Australia you can use it to skip a master's degree and go straight to a PhD. So that's why I'm doing it. It's like instead of taking 2 years to do a master's, I can just do 1 year, an honours degree, and go straight into it. And the other thing is it lets me do a research project with very little, I guess, control and requirements on me. All I have to do is show that I can, do some basic research. So it lets me do something that I probably couldn't get funded anywhere else. So, so here's this opportunity. I've got access to one of the world's largest supercomputers, and I was able to. I dug through research to work out what lipids are in a mitochondrial inner membrane, and then created this little 15 nanometer by 15 nanometer patch of this membrane, put it in and configured it for the supercomputer inside a little box, and then evolved that system using Newtonian mechanics over 15 microseconds. So essentially, I think it's 600,000 little time steps, and at each stage, the system evolves by the action of forces upon all of the molecules in these lipids. And at the end of that, I can then see all of these molecules moving about, clustering together, forming curvatures. I can look at like the area per lipid and other biophysical properties. And then I can characterize what the biophysical properties of this membrane are. And this is something that's almost too small. It's too small to see really with an electron microscope. With all of the tools that we've got these days, it's too small to be able to see. So as far as I'm aware, this is the first time it's ever been done with a complex membrane. I believe it's been done with a very simple membrane with just 2 kinds of lipids, but this is the first time it's been done with the complex membrane with 60 odd lipids. And I believe I may be the first person to have ever witnessed an inner mitochondrial membrane at molecular scale over a biochemically relevant time period.
Right. I think that's a really cool mantle to be able to claim. I'm jealous.
I'm going to wear it.
Right, and rightly so. Yeah, I think being able to characterize something is really important to start to be able to talk about it with a common language. And I think that's why it's a pretty important step, because at the moment we have a bit of a cartoon view in the pejorative sense of the mitochondria. And yet I think there's a level of complexity here that you're starting to scratch the surface of. 60 different species and quantified as well. So you can give an idea of which species is more dominant, maybe. Does it change according to diet? Those are some of the next questions you mentioned you're going to explore. And that's like real basic science, but it has huge implications, I think, for cancer. That's the first thing that comes to my mind. And of course, just an understanding of metabolism more at the more basic level, because we have this proton gradient that's affected by the membrane. And membrane fluidity.
The membrane is responsible for it.
Yeah. Right. And appears to be really important, but we don't know why yet. So I think that's really cool that you're taking research in that direction. And maybe you can say more about that, what it makes you think of as the next steps.
So I'm looking for a spontaneous formation of pores in the membrane. So when you think about it, so inside the mitochondria, you have the smooth membrane on the outside, and then you have this bacterial membrane on the inside. And common to bacteria, it has a special kind of lipid that only exists in this membrane. Out of all of the membranes of our body, this one kind of lipid is a special one that's got 4 tails, 4 acyl tails. And most lipids, you may or may not know, have 2 acyl tails. Most of the lipids in our membranes, the lipids in our plasma membrane, have 2 tails. Predominantly phosphatidylcholine, phosphatidylethanolamine, phosphatidic acid, phosphatidylglycerol, phosphatidylinositols, and cholesterol. And phosphatidylserines. They're the majority species. But then each of these species, depending on whether it might have 2 palmitic tails, in which case they're C16 tails. This is very similar to palmitic acid, the fatty acid that is common in our own body fat, or oleic acid, oleic tails. So each of these different head groups, these phosphatidylcholine, phosphatidylethanolamine, and the like, can also have different combinations of species of acyl tails, and they could have highly unsaturated tails, like for example arachidonic acid, which has got 4 double bonds, or a linoleic, for example, with just 2 double bonds. So the makeup of these lipids in our membranes, it basically is driven. We have there's a biological driver of lipid fluidity and regularity, but there's also an environmental driver, and that environmental driver is diet. So the more, for example, if you eat a diet of only oleic acid, for example, it wouldn't be possible, but let's say for the sake of argument you do, then all of your membranes will be enriched with oleic acyl tails. All the lipids in the membranes will be enriched with acyl tails. And so then you can ask the question, if I'm going to simulate this thing, if I have, for example, a diet high in linoleic acid, so for example, safflower oil, am I going to see spontaneous pore formation? Now, the inner membrane is where we set up the proton gradient that we create all of our energy from. So we have all of these proteins that pump, they basically split electrons from hydrogen atoms, and they pump the proton from that hydrogen atom into the intermembrane space between these two membranes. And then when it comes back into the matrix of the mitochondria and rejoins its electron, it drives a little generator, and that generator is called ATP synthase. And it's like a little generator that turns electrochemical energy, the gradient difference between protons in the membrane, on one side of the membrane and on the other, turns that into first mechanical energy, and then that mechanical energy then stamps ADPs into ATP. So it's literally turning it into chemical energy. So it's a little transmuter of energy from three different states, which is the most fascinating piece of micro machinery you'll ever see. And so if, however, a change in diet changes or causes pores to form in that inner membrane, then what happens is those protons, instead of going through this little turbine generator that makes ATP, they go through a hole, this pore that is formed spontaneously in the membrane. And so it's very interesting to look at how stable the membrane is when, in response to different diets. Do pores automatically form? Could those pores leak protons, for example? And so you could then generate a hypothesis that you could then test experimentally to say, well, does the subject or does the test subject experience energy problems when on different diets? And you can then sort of take it sort of further with that. So, it's very interesting. And the other thing that I find fascinating is that the mitochondria has the master keys to the cell. So if the mitochondria decides that it's not able to produce energy properly, then and I can talk a little bit about the actual mechanism, but it is capable of telling the cell to go kill itself. And so cellular apoptosis. So, for example, in the case of tumor development, the mitochondria are very, very involved in that particular process. So there's a couple of hypotheses. One of them is one that I've developed that I hope at some stage in my PhD to test experimentally and see if I can pull apart the skeins of that and see if I can find something meaningful about tumorigenesis and a failure of cellular apoptosis that allows cancer cells to become essentially immortal.
Right, because usually I think it's cytochrome C release that's a signal, a proper apoptotic signal. But when you get things like necrotic cell death, who knows what's being pushed out of the blasted mitochondria first?
Yeah.
So it'll be important to understand that better.
So cytochrome C is one of these proteins. It's part of the electron transport chain of the inner membrane, and it's an interesting protein because it's anchored to the wall of the inner mitochondrial membrane by these cardiolichens. And cardiolichens are an interesting lipid species because they have a charge of -2, which is unusual for a lipid. Most lipids are either +1, -1, or most of them are neutral charge, but these have a charge of -2. So you could imagine that if you can build up, if you're able to have a lot of protons in the intermembrane space, so you have a lot of these positively charged atoms in the inner membrane space, then that pushes those cardiolipids into the inner mitochondrial membrane, and because the cytochrome C is anchored to them, it stays in place. Now, what happens if, for example, the level of charge in the charge gradient changes, and let's say there's a problem with your mitochondria, it's not able to generate energy, all of a sudden it can't push these positively charged protons into the intramembrane space, then all of a sudden these cardiolipids have less electrochemical pressure pushing them into the membrane. If they then release cytochrome C, it is simply the mechanism of releasing cytochrome C that starts the whole process of cell death. So that's how a mitochondrion can, when it's not running properly, can say to the cell, okay, this is impossible. I think we need to send everything back to the knackers. We need to recycle the cell and turn it into a new cell.
Yeah, because we have this ability to recycle ourselves to a large extent, which is remarkable. There's a lot of energy that's going into turnover, which is essentially just I don't know, maybe it's the natural decay, but it's also the natural cleaning that accompanies it, that's constant and that's useful, because it allows you to react fast. There's this thing about flux, and I think Dave has talked about this in his energy model, that when you have flux and when you look at the so-called futile cycling of fat, actually it's allowing you to be very much responsive, because when it's like a car, you don't want to start and stop your car every 2 minutes to accelerate or change direction. You want it to be on and constantly turning and be able to upregulate its speed very fast by just pressing on the accelerator. And we have the same need, I think, in our cells and in our physiology more generally at the level of lipid cycling. So this rapid response is really important. And so being able to enter processes like autophagy, which is not one thing, but it's a bit more complex than one thing, but it's important. There's a cyclical nature to it, and there's you have to, it always happens to some degree, right? It's happening in the background, then it's upregulated and so on. And I think, we need to understand that at a better level. So I'm glad you're exploring that.
Yeah, I mean, there's also mitophagy, which is the eating of mitochondria, and also mitogenesis as well, which is the creation of new mitochondria. So this is also all happening all at the same time. This is one thing I learned in biochemistry at uni, that nothing really turns off or on. There's very few binary switches in our systems. That it's more up and down regulation of systems to change rates of things happening, and that's really how our processes work. I think the only really binary switch that I know of is insulin, the response of lipolysis in adipose tissue to insulin. I mean, that really goes off the cliff. The asymptote for that is just off the wall. So that almost is a binary switch. But most other things are gradients of rates turning on and rates turning off, and lots of. I guess one of the things that sort of fascinated me about biochemistry, and the reason why I switched from software development to biochemistry, was it's a bigger puzzle to try and work out, because you have all of these homeostases that are all running at the same time, and it's the interplay of those, the dance of these homeostases, that tells you what the phenotypic expression of a genotype will be. And so that's fascinating, is to see how all of the interplay of all of these forces that are acting upon very simple molecular machinery. It really is a fascinating field to be in. And you brought up the word homeostasis, which triggered me into bringing up an old comment that you made on your two Keto Dudes podcast. I think it was in 2020, where you were talking about Professor Leibel and his views of what happens to basal metabolic rate.
and how you interpret the data differently than him. So if you don't mind, I'm going to read it and I'm going to ask you to then
sure.
expound on it, because I found it really interesting, and I had posted it in the Nutrita group.
Okay.
Yeah, go ahead.
Thank you for that. I should mention I didn't know what I was talking back then, so.
Perfect.
Well,
Perfect.
Let's see if I did know what I was talking about. Go ahead.
Actually, it was from 2019. I just seen that. So you say no one really knows why the metabolic rate of people who lose weight on diet slows down. Professor Rudolf Leibel did a famous study showing the effect. They took people and reduced their caloric intake by 10%, and their metabolic rate dropped. Half the people, they reduced their calories a further 10% and saw a further drop in metabolic rate. The other half, he returned them to their previous intake, and their metabolic rate returned back to baseline. Then he took some and increased the calories they ate by 10%, and their metabolic rate went up. Most people, including Professor Rudolf Leibel, took from this that the body was defending its preferred weight, which would mean all diets are futile. I don't agree. I think we've seen experimental data in Dr. Ludwig's recent paper showing low-carb dieters see significantly less metabolic slowdown, and anecdotally, we've seen thousands of people on ketogenic diets lose significant amounts of weight and keep most of it off for years. That suggests our dogma may be wrong. I have an alternative hypothesis, and it is that the body determines metabolic rate as a function of available energy, which is total dietary fuel plus stored fuel multiplied by the access to stored fuel multiplied by the ability to convert fuel to energy. On a low-carb diet, we increase both access to stored fuel, quote unquote, and ability to convert fuel to energy, quote unquote. That is why I went back to uni to study biochemistry, and it will be the area I intend to research for my PhD.
So I guess I've changed my area of interest. That still interests me, and I don't disagree with any of the comments made by Richard Morris in the past.
Talk shit to him. Go ahead. He won't mind.
Yeah, come at me, bro. So I think my area of focus is going to be mitochondria, because that's where a lot of the action happens. But one of the stories I can tell about the mitochondria in the context of this particular example is that when we need to get fuel into the mitochondria, because the mitochondria is what's going to turn fuel into energy. And the fuel could be, for example, proteins, amino acids. It could be pyruvate from glucose, from carbohydrates. It could be fatty acids from triglycerides, from fat. And so these three sources of these molecules that we can basically stamp out, we can basically split apart these molecules, push protons into membrane space, and then stamp out new ATPs. This is how we make energy. So one of the interesting things is that not all of those molecules get into the mitochondria easily. So there are transporters specifically for amino acids that can, because the mitochondria has as I said, it's got its own DNA. It's producing its own RNA. It's transcribing its own RNA, messenger RNAs, and it's essentially making proteins inside the mitochondria, just like a little bacteria sitting inside your cell. And so you need to have the raw materials for that. So you need the proteins coming in. Now, pyruvate has a transporter specifically for getting the raw material from glucose into the cell to be able to be converted into energy. Fatty acids, however, the short chain, because the membrane is essentially a fatty, it's aliphatic molecules. These are molecules where the outside head of the molecule is hydrophilic, so it quite happily sits in the aqueous solution of the cytosol, but the inside part of it is hydrophobic, and so these self-organizing structures of the lipid bilipid phospholipid bilayers that form these membranes, the inside is aliphatic. The inside is hydrophobic, and so all of these molecules that are like pyruvate and amino acids won't go through, because the only thing that will go through is short and medium chain fatty acids. So they go through they diffuse through the membrane quite easily. So if you eat, say, a short chain fatty acid like butyrate, which you get from butter, that'll go straight through the mitochondria, not a problem. You can easily burn this. And no matter how much butter you're eating, you're probably going to be able to get it into your mitochondria. There's going to be no barriers to access, and you're going to be able to burn that. Medium chain fatty acids like, for example, lauric acid, caprylic acid, basically I think C8 through to C10, C12/C14. C14 is sort of on the border between medium and long chain fatty acids. Those can all go through. They can diffuse in and be converted straight to energy. So that's great. If you have coconut oil or butter in your diet, you have no trouble burning fat. The problem happens with anything longer than 16 carbon chains long. That has to be actively transported through a transporter called carnotyl palmitoyl transferase, which is an enzyme that sits on the outside of the mitochondria, the outer mitochondrial membrane, and it transfers. In fact, there's a couple of proteins that do this, and they essentially combine. They coordinate together to transfer, basically conjugate the palmitic acid, the long chain fatty acid with CoA moiety, and then they transfer that into the cell. So essentially, it's a transporter on your mitochondria that's basically pumping the gas. It's getting all of the energy from fatty acids into the mitochondria to be burned for energy. Okay. So one of the things that happens is that I'm going to introduce insulin. I probably shouldn't at this early stage, but let's forget about insulin itself, and let's talk about just insulin signaling. Hormones that signal our cells do a bunch of things. They can turn on or turn off enzymes, either phosphorylating or dephosphorylating the enzymes, and sometimes that turns them on, sometimes it turns them off. So it can be confusing, but as I said, it's an interesting puzzle to solve. So there's this enzyme called acetyl-CoA carboxylase, and it is the first committed rate step for making new fatty acids from, if you eat a lot of glucose, you're going to make new fat. You know how that works. If you eat too much glucose, then you need to immediately burn and you can't store anymore. You're going to make it all into fat. So we know how that works. So the first enzyme that does this is this ACC, acetyl-CoA carboxylase. Well, it turns out that insulin signaling phosphorylates that. So it turns the enzyme on. So the enzyme's just floating around the cell. If it's not phosphorylated, it won't do much. But if it's phosphorylated, immediately it finds acetyl-CoA, it starts turning it into malonyl-CoA, which starts the process of making new fat. So insulin goes high, start making new fat from acetyl-CoA. The other thing that it does, the insulin signaling, is it promotes the transcription of new copies of this enzyme. So not only does it turn them all on, it makes more of them. So and this makes sense in a evolutionary context, because when insulin goes high, you've got a lot of glucose coming in your diet because glucose-stimulated insulin secretion has gone up, and so you have more insulin signaling. And so if you've got lots of glucose pouring into the cell, you want to be burning glucose, and you want to be making fat. And the other thing that the insulin signaling does is it tells the body to store it as whatever it can, causes cells to store some of that glucose as glycogen. So the interesting thing here is that when insulin signaling goes up and it starts this process of making this enzyme make more malonyl-CoA and eventually to make palmitic acid to be then exported, it turns off that transporter, carnotyl palmitoyl transferase, that gets long chain fatty acids into the mitochondria, because there's no point in burning fatty acids if you're making fatty acids. You want to either be doing one or the other. Well, it turns out that if your insulin is always high, if your insulin levels are always high, then rather than import it and being able to burn fatty acids, all of a sudden now your mitochondria aren't transporting them in. And so the fatty acids build up as lipid droplets into cells, which we can see as part of the cascade of intramyocellular lipid droplets that cause cellular insulin resistance. And of course, if you've got fatty droplets in the cells, then your transporters that import fatty acids into the cells from circulating lipoproteins downregulate, and now you've got triglycerides building up in circulation. And that explains part of the mechanism of why type 2 diabetics who have insulin high all the time have a lot of fat in circulation. It's nothing to, it's nothing really to do with overflow from fat from a what do they call the fat limit, the personal fat threshold.
Personal fat threshold.
It's not any. Yeah, it's actually the fat threshold is the carnitine palmitoyl inhibition by malonyl-CoA. So it's
go convincing people of that.
But it's not. That's the thing is,
right.
we tend to think it's kind of it's the source and the sink problem, really, isn't it? What we observe is the amount that's floating around in circulation, and we see all of these people with, like, when I was a frank type 2 diabetic, I had my triglycerides in circulation were 1,111, which essentially meant that in my 5 liters of blood, I had 55 grams of fatty acids transporting, unable to be used. And the reason why they're unable to be used is because of this inhibition at the mitochondria. It's kind of like if you had, let's say that your fat cells, let's say that your circulation is a bathtub. And we're filling the bathtub with fat cells. And so how does that bathtub get full? Well, it gets full from there's a couple of spigots, a couple of taps. There's you're getting some from adipose tissues releasing fats. We've got some coming in from diet. We've got some that we're making. So we've got these three sources of fatty acids in circulation that are happening. So how does it get out of circulation? Well, it gets out of circulation by being used. So it's essentially the drain of the bathtub is your mitochondria taking these things and turning them into ATP. So what if the mitochondria isn't able to do that because of this inhibition, this rate inhibition? So it's like having a face washer. You've got a face washer in the bathtub, and now you've got these 3 spigots, these 3 taps of water pouring water into the bathtub. What's going to happen? Your bathtub's going to overflow. What happens if you just turn off one of those? Let's say you stop eating fat altogether. What happens? Your bathtub still overflows because there's still 2 taps filling it with fatty acids. The only way to fix this is to fix this rate inhibition, to take this face washer out of the drain and allow the water to go through, and then your bathtub never overflows. So, it's a fairly simple process. In fact, there is in the literature examples of diabetic rats, for example, that the rate constant of inhibition for malonyl-CoA is 50 times higher than that for non-diabetics. And so these diabetic rats are making fat all the time, and they have to have this inhibition. The rate constant of inhibition needs to be raised just to enable some of the fatty acids to get in, because they're being made all the time. So, so essentially, in a metabolically deranged animal, you've got they're actually making new fat from old fat, and they're making new fat from carbohydrates, and they're making new fat from excess amino acids, and essentially they're making new fat from everything. So that's a highly deranged, and that explains essentially why type 2 diabetics become obese well before their glucose starts to rise. It explains why they have high triglycerides. There's no other real good explanation for why a type 2 diabetic would have over 1,000 milligrams per deciliter of triglycerides in circulation, and unless you can explain why it's not being used, and that's probably the explanation. So that's all at the mitochondrial level. So,
I think ultimately it does come down to what the mitochondria is going to be able to handle, in a sense. And I think what you just explained also goes to resolve an apparent paradox called the athlete's paradox, where we see a lot of intramyocellular triglycerides in the muscles of type 2 diabetics, right? They accumulate a lot of this fat. And we see it in athletes, which, the notion would be that they're not type 2 diabetic, right? They're considered the healthiest. And then the normal people, which are not athletes, but not type 2 diabetics, have low levels of intramyocellular triglycerides. And I think that can be explained by the rate of lipolysis. So in the athletes, they're going to have a high rate of lipolysis because they're using a lot of fat efficiently, and you have a lot of use of that triglyceride in the muscle as well at the same time. And the type 2 diabetics is, yes, also going to have a high rate of lipolysis, but they're not able to use that muscular fat as well. Their mitochondria aren't able to use it, so it accumulates. So, and of course, the normals have a low rate of lipolysis and a low rate of use of triglycerides, but they don't see this accumulation. So I think it could explain that as well, indeed.
Yeah, I have a paradox that I call the hungry fat man. And I'm in the process of writing a book about this. The paradox of why is a fat man, somebody, an individual who evidently has plenty of energy, why are they hungry? What explains why a fat man is hungry? What explains why he's parsimonious with energy? Why is a fat man greedy and slothful? The only explanations that we really have had to date is, it's a moral issue. It's a moral crisis. They're weak, and we're feeding them horrible food, and it's processed food, and we're giving them something magical in their environment that's changed, linoleic acid in their environment, or too many carbs. Those are contributing, to the process. But the reason why the fat man is hungry is because he's unable to turn his fuel into energy. He's unable to turn fuel into ATP. He's only able to turn fuel into new fatty acids. And so he's locked in that cycle of making new fatty acids all the time. They never get to be pumped in as protons into membrane space and converted into ATP, because they're being greedily used by all this acetyl-CoA carboxylase that's being created. So that's my theory, and it explains the paradox, and it also probably goes to explain this athlete's paradox. But it's So I think that's probably the simplest explanation of that.
It has implications for a lot of diseases. How we're able to use fat certainly seems to be important for diabetics, and it seems to be important for Alzheimer's disease. Although it has to do with the complex 1, not being able to bypass complex 1 in Alzheimer's disease. That's probably why ketones are so useful.
Is that why it is? Explain to me about Alzheimer's disease.
I think so, because it seems that those populations of cells that should be able to burn glucose to sustain themselves, when that is broken, when it doesn't happen through complex 1 as it should, you need an alternative, and the ketones allow you to bypass it. It goes through. There's the flavoprotein transporter, and I think it goes through
F80+,
which is it? Complex, F80+. So you can bypass it, and that's why you can see some very quick improvements in the cognition, because the machinery is already there. Really, there's no big upregulation needed to do. You know how they diffuse across the blood-brain barrier beautifully. There's no impediment there.
They're small hydrophilic molecules that travel by themselves in the bloodstream. They don't need to be carted around the place with carrier proteins, and they're so they avoid complex 1. It's interesting because I know that metformin inhibits complex 1. That's one of the things it does. In high concentrations. And it's interesting how metformin helps people with type 2 diabetes, because especially if they're on a high-fat diet. If you have somebody with type 2 diabetes on a high-fat diet who's then their glucose homeostasis gets a lot better. If you then get that same person on the metformin, it performs even better. So it's like a synergistic relationship with a high-fat diet where they're running on a lot of ketones. That they don't. It's interesting hypothesis. I need to drill into a little bit more about the distinction between complex and the other.
Although what seems more relevant mechanistically for metformin seems to be the interaction at the level of the glycerophosphate shuttle. And because I think the concentrations at which the complex 1 interactions happen are like crazy, like super physiological, basically. So at the levels that you'd see it circulate from a pharmaceutical in the blood, I think it's usually the most likely point of interaction is the glyphosate shuttle.
Right. Okay.
So yeah, so just it'll be interesting for you to look into that. I think Peter's written about that probably the past year. So there's an old blog post there to be dug up.
Excellent.
Yeah. So let's see. Is there anything just to jump off to something different? I was wondering if there's anything in the current news, whether it's diet wise, COVID wise, science communication wise, whether it's something to do with your research, something that's taking your attention. Is there something?
I'd like to get into more science communication. I really should, because I did enjoy that part of Keto Dudes. But I've been tempted to do, I've got a green screen behind me, and I've been tempted to start doing some YouTube videos explaining mechanisms, for example, how some of the vaccines work, for example, the mRNA vaccine. Fascinating, fascinating story. I mean, did you know that they actually had the vaccine, the mRNA, the Mrna mRNA vaccine that is going into people's arms right now was completed. Its development was completed in January of 2020. Wow! And everything since then has been just safety and efficacy and dosing.
That's amazing.
It's remarkable. I mean, it literally, as soon as they had, I think it was very late in January, maybe the 28th, when the genetic sequence for the spike protein was first published. I think it was published by a Melbourne lab, and I think within a day or two, what would eventually become the final Mrna vaccine was created. So it's just remarkable, and everything since then has been just making sure that it doesn't kill people, it doesn't have weird side effects, with a large enough audience. So there's that.
I can't remember who, but wasn't it a woman who had been pushing the theory of a vaccine that's built in a ribosome, an mRNA vaccine that's built in a ribosome to really create a specific immunological a target, basically an antigen that can allow you to create an immune response without creating a lot of let's say side effects, like a lot of cross immunity to other stuff and an exaggerated immune response. And I'd read that for years she'd been trying to publish the concept, and it'd been very difficult. And finally, we see it created so fast. That's really cool.
It's a triumph of molecular cell molecular biology. It really is, because I did an assignment in my final year at university on I was asked to come up with reasons to support genetically modified organisms, GMOs. And one of the ones that I came up with is that the distinction between, let's say we've got a wheat variety, and we want to make it more resistant to say being blown over by wind. We can look through its genome and work out what genes are required for extending the proteins in its stalk, and we can modify those genes directly, and we can then produce that variety and see if it has that effect. So that's the GMO side of things. The non-GMO way of doing artificial selection is essentially we go out and we plant a bunch of wheat, and we see what wind knocks over which plants, and then the ones that aren't knocked over, we grab their seeds and then we assume a random mutation. We might actually irradiate them, for example, to push mutations. And then once we find a mutant variety that produces the desired behavior, we then cultivate from that variety. But the difference between GMO and artificial selection is that artificial selection is a scattergun approach. We're not targeting directly the thing we know that's going to cause the behavior that we want. What we're doing is we're rolling a dice and picking the one that wins. But there might be other things that have happened. There may be other mutations that have happened to that particular variety. For example, what say after 30 years of eating it, somebody becomes, the gluten in it damages the zonulin borders of their gut, for example, and they become celiac develop celiac disease. You know, these are kinds of things, these sort of off-target effects of interventions. A GMO allows us to skip that. So it allows us to be very precise about the change that we want. And in the same way, by in the old days with the vaccine, what you do is you take a version of the virus, and you immobilize it somehow. You make it less able to procreate, to reproduce. I shouldn't say procreate, because of course they're not alive. But you make them less likely to reproduce, or reproduce at a lower number that can be easily overwhelmed by our immune system. Or you make them, for example, there are some the ones that they get spray in your nose, for example, they reproduce quite well at room temperature, but not at body temperature. And so in the nasal cavity, they're going to reproduce. And then as soon as they get into the rest of your body, their reproduction rate drops dramatically, because we've attuned them somehow so that they're more susceptible to temperature differences. That's the old way of doing it. But what you're doing is not only are you giving the body some antigens to express some antibodies to, but you're also giving them all of the payload of the rest of the virus, the rest of the stuff that could potentially cause problems. And so there's lots of off-target effects. I mean, you're rather than giving the vaccinee just the spike protein so that they can just make an antibody for that, you're giving them all the proteins of the virus. So it makes all of these, and every single one of these proteins, these antigens that you're going to make an antibody for, there's a potential for autoimmune problems. You know, you never know when your body, you walk into a strange room and you get a strange antigen. You don't know what it's going to be. It could be from a virus, could be from a dust mote, it could be some pet dander or whatever, and your body makes an antibody against that particular antigen, and it thinks it's really clever, but it turns out that antigen is very similar to the shape of a part of your body that's very important. And now your body is going and, let's say, removing your beta cells that create insulin, and now you're a type 1 diabetic. So you know, the more sort of targets that you give your immune system to respond to, the more likelihood there's some off-target effects. And so one of the nice things about this mRNA vaccine technology is it's just the spike protein. Now, if it's going to cause a problem, then the virus will also cause the same problem. So you know, if it's going to cause any problems that are likely to happen are likely significantly worse with the actual virus itself. So you're immediately in a better position. But you're also just targeting the things that are on the outside of the virus that the immune system is going to come into first contact with. So you know, and there are two ways of essentially vaccinating somebody. If you're not going to give them a live or a dead or attenuated virus, then there are two ways that you can essentially vaccinate someone. You can give them the antigens, give them the proteins. You inject the proteins into them, and they make antibodies, and eventually now their immune system is ready for the actual virus when it comes along. Mm-hmm. Or you can give them instructions, a little recipe, if you will, so that their cells can make lots and lots and lots of copies of this. And so you need to give a much smaller dose. You just need enough mRNA that's encapsulated in these lipid nanodroplets to be able to get into cells, to be able to start making these proteins. And now your cells can do all the work of scaling up the factory. So if you're going to give somebody, you know, the antigens as proteins, you have to give them a lot to be able to get, you know, immune response. Mm-hmm. But in the case of the mRNA, you have to give them a small amount, and what happens is that mRNA goes into your cells and starts being transcribed into the protein, and now you're making this antigen that is very similar to the shape of the spike protein, and now your immune system's getting used to that. And what will happen is your immune system will take out the cell that has been making this protein. So it's a short-term thing. It's a self-fixing system. So you know, it's a very clever technology, and all we need to do is sequence the virus, and we can pretty much have the mRNA vaccine the next week. You know, it doesn't take that long to create the mRNA necessary to produce a protein. With that,
it's really a step function improvement if this works as we think it'll work. I mean, that's really cool. And it's a pity that we can't actually focus more on that, because we're in such a terrible time for all sorts of reasons. And it's kind of a pity, because as much as I'm so enthusiastic about what I think it can do for the reasons you just gave, I'm very much unhappy with the whole program around how it's been developed. How it's been tested, I should say. Because as you said, actually it was developed really quickly, but then we've just been testing it for, nearly close to a year now. And
yeah,
now
each of these
being
vaccines have
out.
had each of these vaccines has had 40,000. It's gone into 40,000 arms, or at least it's gone into 20,000 arms, and 20,000 arms also had the placebo, the same protocol.
Right.
So 20,000 is not a large number to be able to extrapolate how it's going to affect when you put it into 7 billion arms. And so that's, it gives you an idea. It's going to get, it's going to get sort of 20,000 out of 7 billion. Is a pretty, it's not nothing, but it's because we're in dire straits. We need to be able to come up with a treatment for this to be able to drive herd immunity, because we're in an evolutionary race now, because every single host, every single human being that's been infected with this virus for at least 14 days and probably 28 days, that virus is replicating in their system, and eventually it goes to find another host. If the virus wins, the virus gets another host. That's how the virus wins. If we can prevent the virus from getting another host, that's how we win. But every time it gets into a host, it's going to replicate several billion times, and every single one of those billion replications potentially could find a mutation that makes them easier to transmit, easier to infect, be able to become more endemic in the human population. So, countries like America, I mean, it's frustrating to me how poorly America handled this. I mean, they have 250,000 new hosts every day that they're adding to the pool of potential. We have a lab, and the virus has a lab, and both of us are trying to compete with each other to wipe the other one out. Well, the virus is probably not trying to wipe us out. It's probably trying to become, part of the, endemic in the human body, like the other coronavirus. And the every time a new human is infected with this vaccine, every new host is a new lab, or potentially billions of labs for this virus. And every opportunity is, I mean, the chance that the virus will mutate with 250,000 new hosts every day is so much greater than if we were able to, keep a lid on this thing. So it's frustrating to me.
Yeah, I think the only question really is, how do you keep a lid on that? And I think that's where, I mean, we know whether through what, I mean, look, in when we had SARS-1, it eventually disappeared, and we don't know why.
No, we don't.
Because everything you've just said is valid for SARS-1, and yet it disappeared without a vaccine. So the question is, what is it how can we win in this war of our lab and its lab? Yeah. And I think there's a lot of unintended consequences to that strategy. So if we look at the measles vaccine, I mean, it's an amazing success story. Just to give you an idea of the number needed to vaccinate for measles, it's 1.17. That's insanely effective. You need to save someone from the potential of getting measles. You don't even need to vaccinate 2 people, just a little more than 1.17 people. That's astounding. Now, for the coronavirus, based on the data that we have now, which is that it's 90% effective in reducing transmission, which is defined as a PCR positive test protocol not offered, but or one or more positive symptoms and a positive PCR. So based off of that, it's 95% effective to prevent transmission. Now, translated from this relative risk into absolute risk, if you compare it to the measles vaccine in terms of number needed to vaccinate, for a severe case of COVID, you would need to vaccinate 1,370 individuals
to save
to prevent. Yeah, say one to save someone from a non-serious case of COVID, but a case of COVID nonetheless. It's 176. So it's interesting to me, because I think it it tells us that there's so much more to a vaccine than the vaccine itself. There's the population it's given to, the susceptibility, whether it's by age or by comorbidities or something like that, at which point in the epidemic you're giving it, how close are you already to herd immunity? Because then it has more or less of a job to do for you in that sense.
That's
so
right,
your numbers are going to change. And I think it brings in this whole idea that I actually love, I have to be honest. It's not vaccines that save lives. It's vaccine programs, given at the right dose, at the right schedule, by the qualified people, at the right point in the pandemic. All of these things, I think, go into it. So that's where I come from in the vaccine question. I'm in awe of the technology. I'm just kind of in despair to how it's been communicated and how we're going to implement it in the end.
See the difference between SARS-1 and SARS-2 is that SARS-2 has this really long pre-symptomatic infectious stage. So it's like something like 4 to 5 days before your symptoms that you're infectious, and so we can't tell who's at risk. Whereas SARS-1, they got sick first, and then down the road they got infectious. And so by the time they were sick, they were in hospital, they were in isolation. So it changes the dynamics of that particular picture. And that's why I honestly believe that SARS-1, the reason we got on top of SARS-1 was because it advertises its presence way too early in the picture. This bugger is sneaky. This bugger doesn't advertise its presence until it's already infected other people. So, or already had the opportunity to infect other people. So that's why there's a difference in the dynamics of the two viruses. But as far as the number needed to treat to save one person, I've always found that difficult to come to grips with the math involved with that, because it's essentially it's not just the vaccine that's going to be fixing the problem. The problem that you're trying to fix is denying the virus a host. And the vaccine is just going to be one extra barrier. It's like a condom preventing conception. I mean, 99 times out of 10, out of 100, the condom will prevent conception, but the condom plus the rhythm method is guaranteed, is almost 100 times out of 100. So it's one of these things where it's not going to be enough for enough people to be vaccinated, although it will be good. We are going to end up changing. In the same way that September 11 caused us to have to take our shoes off and our belts off before getting on a plane, which sounds ridiculous in 2000. Pre-September, on September the 10th, 2000, if you were to tell me you're going to, in the future, you're going to willingly take your belt off before you get on a plane, I would have told you you're crazy. I think we're going to change the way that we think about the potential transmitting viruses, in the same way as Asian populations after SARS-1 started wearing masks more often and having more social distancing. And to be honest, they had less of the problem with SARS-2 because they'd already developed these natural human behavior based strategies for limiting spread of viruses. So we're lucky in Australia because we get 3 new cases a day and we start to panic. We're going to not play a game of cricket, or we're very close to not playing a game of cricket because we're getting 3 or 4 cases a day in the entire state of New South Wales with 4 million people. So we're in a very different situation than the rest of the world because we're in an island out the back end of nowhere. We have the same advantage as New Zealand. We were able to shut off all the traffic coming in, but also we need the vaccine to be able to go back to normal, to in any semblance of normal. For people to be traveling in and out of Australia, we're going to need to have the vaccine. And I do think that we're going to change our human behaviour to make it a little bit more difficult for viruses to transmit from host to host. And that's probably going to be good for us for influenza and a bunch of other respiratory viruses that plague us, kill a lot of humans every year. So I think in general it's going to be a good thing. But I take your point that you know, the measles vaccine is remarkable because the measles virus is remarkable. You know, it's a dry respiratory virus. It's you know, it doesn't need to be in a it doesn't need hydrophobic collapse to hold its envelope together. You know, one person with measles walks through a room, and you know, the 18 people in the room get measles. You know, within 18 people walking into that room within 30 minutes later could 18 out of 100 could pick up measles. So you know, it's a very different category. But yeah, but I do take the point. I mean, I think we're going to have to change. I think it's going to be a major change for us.
Well, I think we're going to change. So I agree with you there. I don't think that means that it's necessary to change as we probably are going to. I don't think the Asian model is a particularly successful public health model upon which to base ourselves, because I think the elephant in the room is the peril we put ourselves in with metabolic disease and all the things that we usually talk about. And I think that hand hygiene is common sense, but it's also very minor in its effect. It just happens to be a small effect. I don't believe much in the public use of masks just to quell the transmission, but I do think that it's probably worth doing in a very targeted population who know how to use an N95 in certain settings. So that's where I draw my line personally. I wouldn't. Sure. I say if you want to wear it in the streets, go ahead, but I wouldn't make it mandatory. And so all of that being said, it's really a question of if we pin our hopes on the vaccine, which I think now it's inevitable. That's whether you think it's right or wrong. I think that's what's happening basically. My fear is that the vaccine is not the vaccine is going to make the technology look bad. And it's not because of the technology. I think it'll be because of where we are in the epidemic, how it's being rolled out. We're seeing in the UK now, they've delayed the schedule, which means now you've added a selection pressure to create an antibody response where you're not going to get the optimal amount of neutralizing antibodies. Which means you're going to, and there's all these things where I'm like, God damn it, we're doing it again. We're just doing it again, you know? And it's a pity because, we're half-assing it. And go ahead.
It's like putting antibiotics into soap, you know.
Right.
We're creating selection pressure to create antibiotic-resistant germs. I think in the case of the UK, I understand why they're doing it. The reason they're doing it is because when they did the trials, the reason that they say that these AstraZeneca is 65% effective is because if they had two groups in the trials. One group had saline, or they probably had an inactive vaccine put into with adjuvants put into another similar kind of vaccine, but not for this particular virus, so that they could have a like-for-like comparison. And for every 100 people on the placebo arm that got the virus, 100 minus 67 or 65 on the treatment arm were infected. But the interesting thing is 100 of the 100 people had a case so mild that they didn't have to go to hospital. And so they're in a position now where they're trying to protect the National Health Services, and they're looking at a bigger problem than they had in April. And it really comes down to do we take the risk, the unquantifiable risk of giving selective pressure on non-neutralizing antibodies for this vaccine to this virus to potentially cause it to find a mutation that escapes the vaccine, or do we keep enough people out of hospital that the whole system doesn't fall apart? So I can understand why they're doing it. They really must have a real shortfall of production for it to have to be 12 weeks. So I think it was only like 2 weeks, wasn't it? The normal. I think
it was there about 2 weeks.
2 weeks. So they must have had a major shortfall of production for that to be the case. But the nice thing about the mRNA vaccine is that you don't have that kind of shortfall, because the cells are doing all of the work of the scale up.
Right. Yeah, that's true.
You know,
Again, it's amazing. It's science fiction level cool stuff. I think it's really cool.
Yeah.
But it's always the human level stuff which comes to ruin the party.
The interface between humans and technology is not a clean one.
Yeah, it's funny because, you know, from depending on where a person is coming from, they could easily see my strong criticism of the trial and the endpoints and the release of the data and the science by press releases as someone who's very vaccine negative. But yet I'm also I'll be the first one to complain about lockdowns and the fact that it's going to create, you know, 400,000 missing vaccinations for kids with tuberculosis, which we have a really good, you know, handle on, and 6.7 million extra kids are going to go hungry this year. And there's all these things that just, I don't know, because of the way I'm wired, basically sort of way heavier in my mind. Yet, you know, I'm still rooting for the mRNA vaccine to work because I know how people are and how very old and sick people are in elderly homes where no one's going to give them a low carb diet. It's not going to happen. So if you can give them vaccines, let's do it.
Yeah, that was one of your comments about the metabolic health of people. I totally agree with that. I mean, when you get the vaccine, you need to be in tip-top immunological health because you need to make antibodies. You need to make antibodies efficiently and quickly for that antigen so that you're ready when the real thing comes along. I mean, a vaccine is essentially like a wanted poster being put up of, you know, some bad fella's going to come into town and bank robber. So we put a wanted post up around so that we can recognize him very quickly. And so that's essentially what's happening there. But you want your immune system to be very efficient. You want to not only go low carb and energy sufficient. Do not be well, you know, people are welcome to be calorically restricted for their cosmetic purposes.
Jesus saves.
Knock yourself out. Yeah, I mean, be calorically restricted and develop a six pack during Armageddon. Knock yourself out. I don't care. But for the rest of the world, you don't want to be energy insufficient. You want to be energy sufficient. You want to be eating adequate calories and being metabolically healthy as you possibly can. I don't know if you noticed, but one of the things that seems really common amongst all of the pictures of people who subsequently die very quickly of the virus is they have no jawlines. They've got all that fat deposition under their jawlines from you know, metabolic derangement. You know, that seems to me to be so obvious that, you know, so, you know, if I'm certainly going to be low carb, I always was going to be low carb, but you know, I'm certainly advising my family who aren't low carb to get as metabolically healthy as they possibly can, because you know, this will be important.
And I can speak anecdotally to what you just mentioned, because in my family, I've had coronavirus. My brother, his wife, my dad, and my mum all got it over Christmas. Everyone's fine.
You buried the lead. I didn't know you had coronavirus.
So I think I came home from Portugal pre-symptomatic. So I came home on a Sunday. A friend picked me up. On Monday, I saw my parents, and, you know, I woke up with like a little scratch in my throat, but nothing that I would call sick. And in the evening, I came home, and then I felt tired and kind of feverish, and my little throat scratch had developed into something a little more. And I didn't feel too great for about 2-3 days. I was colder, I was eating less, but that's it, really. And then when I started feeling much better, on the 4th day, I would say, then I started, you know, going running again, but I was very slow. Right. And then I got this total loss of smell one day, and it started coming back 2-3 days later. It came back really fast. Actually, I was surprised. Yeah. My girlfriend, it still hasn't fully come back yet. And it has been like 6 weeks for her.
Carl was infected in March, and he still hasn't returned.
Wow.
Yeah.
It's just the variation is interesting. It's come back from my parents as well. My dad is 73. It was no biggie for him. A few raspy throats and a bit low energy, but that's it. But again, my parents are unusual because they eat a great diet, and they've been doing it for a handful of years now, even though they're in their early 70s. So for us, it's fine. But my cousin's husband unfortunately died from it. He was 65. Yeah, it's I didn't know him well. I only met him once, but he had serious cardiac issues. He had a very, very serious heart issues, and we know that's metabolic derangement. I mean, we know it.
It is. It's part of the picture. So was he infected from your family cluster or independently?
No, he was. So he lives in Milan. So we had no contact with them. Milan. And my cousin, who lived with him, of course, she didn't catch it ever, living close proximity. But obviously, she's healthier than he is, and a little younger. So this disease that's why for me there's the big untold story here, which is sort of frustrating to me because I know it will never get the attention it deserves, is that we made ourselves so susceptible to a disease that otherwise would be less consequential than influenza, probably. Sure. I think.
Yeah, probably. Yeah.
And it's this whole, because then we would have to admit so much fault. It's just not bearable for the authorities to do this. And it's quite frightening to me seeing that and thinking that we're not going to learn the lessons we need from it. We'll have done great things like develop mRNA vaccines for first time, assuming it goes well. So there's good sides that come out of it as well, but I don't think we're going to learn the key lessons here. I don't think we are.
I think we may learn to behave better as a species, as a group organism, a group organism. I mean, as communities. I think there is a lesson to be learned, and I think we're capable of learning how to behave better towards each other. And for example, I think that I suspect, I could be wrong, I suspect that there will be a reconsideration in the United States of social medicine after this. I think that gets back on the agenda, which I think, and so it's these small things that we're getting. I hope we get slightly better at working as a social organism rather than, and I
hope you're right.
Individuals, but it'll be small steps. It won't be a big leap. But I mean, there's all sorts of interesting things like, for example, the coronavirus apparently has color receptors because it affects people with darker skin who at northern latitudes a lot easier than it affects white people in equatorial latitudes. Again, that's all vitamin D. It's all metabolic. It's all metabolic health. Yeah. This is it doesn't, of course. I mean, the coronavirus doesn't have eyes, so it can't tell the difference between a black person and a white person. But a person who is fair skinned, who is living at lower latitudes, is producing more vitamin D during the summertime and is more vitamin D adequate during winter than somebody with dark skin in the northern latitude. So, that's going to be another interesting thing, is that we're going to be able to look at the data now that we're getting more data in. We're going to be able to look at the data and say, you know what, vitamin D was an important thing. And it's probably not just important for coronavirus. It's probably important for populations across the world. And we need to seriously consider supplementing people with darker skin in northern latitudes and southern latitudes with vitamin D, or we need to be prescribing people to get sun for pre-burning sun exposure. Go out for 10 minutes in the sun, get some sun exposure, and then put your UV block on, or your shirt and your hat and your all these kinds of things. So I think those kinds of things also might be opportunities for us to become a little bit more aware of some of these things. And certainly we're going to look back at this, and we're going to look back at those people who had metabolic disease and what the different differential rates of coronavirus worst case complications were. It may actually help us in the low carb community, because we're going to have data. We can actually look back at this later and say, we were right about that. We were right about ketogenic diets. So anyway,
the
data I hope will prove us to be right.
Yeah, I hope I'm glad that you're more hopeful than I am on what we're probably going to be able to learn from this. So I hope I'm wrong and I hope you're right. But we'll see. I guess if we just stick around long enough, we'll get to see it. But
Well,
yeah,
I
this is
haven't
a
got it
yeah.
yet, so I haven't got it yet. So
Well,
we'll
maybe
see.
you won't have to. Maybe you'll get vaccinated and not
I hope so.
have to. Yeah, I don't know if is there any plan for it getting rolled out in Australia anytime soon? Has it already happened?
Yes.
Or?
Well, we have the advantage that because it's almost non-existent in the country, and it's the same with New Zealand, we don't have to use emergency use authorizations. We don't have to sort of essentially what the US and the UK and other countries have done, and Europe as well, have done is short-circuited the normal process of these things being regulated. So we're going to start vaccinating in March. So we're going to go the full 3 to 4 months worth of investigation by our Food and Drug Administration, our version of that, and then we expect to have 25% of our population vaccinated by the first 6 months. By 6 months after that, so between March and September next year, we'll have 25% vaccinated. You'll have to be vaccinated to fly out of the country. I don't think we're going to have mandatory vaccinations, which is good because I would be very much against mandatory vaccinations. Yeah, but it's important that you probably won't be able to fly. So if you want to fly, you'll have to get vaccinated, but it's not mandatory.
Oh my God.
If you, there may be cases. So, for example, there may be situations where you might have trouble getting your kids in school. You might have to homeschool your kids if you don't want them to have vaccines. So there will be costs involved in people choosing not to be vaccinated. But we've never had mandatory. We've never had compulsory vaccinations, and I think that's a good thing. Yeah.
Okay, I see. I think in in the UK it's already happening. In France, it's not ready yet. Well, I just hope we can handle it with common sense and learn. And I just hope we look at the examples in the past at the programs that worked. And I think we'll see that the minimal coercion is always the best strategy, because I think there's so much blowback from the anti-vaxxers when you have this strong-handed approach and harsh labeling of people who are inquiring. I hope we can learn from that, because it needs to get, we need a certain amount of people to get vaccinated, and depending on prior immunity and how many people already caught it, and that will push us close to that herd immunity threshold wherever it ends up being. And that's important, that we can get there.
I don't think we're going to need to have everybody vaccinated to be able to protect those. Though, I mean, the reason you want herd immunity is for those people who can't be vaccinated, or for some reason they not either they can't be vaccinated or they can be vaccinated and it won't make any difference. So for those people, you want to be able to prevent the virus getting near them by having enough people around them that are vaccinated, so that it just simply can't propagate through the mass of bodies to get to them. But I think we're going to need a lot less than we have, for example, with TB and measles. We're probably still. I've heard numbers anywhere from 20% to 70% have to be vaccinated to get herd immunity, and I don't think we know that until we get to herd immunity. So the claims by people who are, there are some people on our side of the park who are very convinced that you only need 20% to have herd immunity, and I don't think there is any evidence for them.
We talked about that with Gabor. He put it more around 30 to 40% according to the studies he'd seen. I followed a conversation between Chris Masterjohn and Dr. Gomez, I think, where she did her modeling with some assumption for prior immunity, which Imperial College hadn't done. And she had a range which was just under Gabor's, essentially, so around 20%, which is not a popular number given that it started out at 80. So it actually is going to depend on the country, first of all.
Yeah.
So it's not one figure. So.
New York has greater than 20% recovered. So and they still have hundreds of deaths a day. So or up to 100 deaths a day. They weren't as bad as they were originally. So I don't know. I put it this way. I
don't know either.
And I think that's a good statement. I think that's a good statement to leave on. I mean, the thing is, there are things you can know, things you can't know, and things that you can know, and things that
Right.
You'll never know. And I think we need to be able to. I highly appreciate people who say I don't know and I'll find out, or I don't know and I don't think anyone can know. I think those are both good statements to make.
Yeah, absolutely. I think that is a good note to end on, actually. So thanks for taking the time to tell us about your work and just give us your opinions on the science we're hearing about in the mainstream. So that was a ton of fun for me. Let's do it again, and let's do more conversations with other people, so we can have we can choose a topic and dig down on it and go deep. Yeah. So that should be fun.
Come back to me in 6 months' time, because I'll have started my PhD by then. So.
Perfect.
Okay.
Perfect. Okay, let's do that.
Thanks, Raphael.
All right. Yeah. Thanks. Bye.